Okay, today's study tip says, your professor is a resource to help you do well in the class. Ask her questions, make an appointment to see her, hang out with her after class, send her an email. Yeah! Do you know where my office is? Probably not. It's over by the bathrooms in Sequoia. Yes, it's by the Natural Science Museum with the big bear. That's where my office is. That's 103. Some of you hang out after class. Like once in a while, somebody will. That's a good one. Make an appointment to see me before class. Send me an email. That works. I don't know. Are you scared to talk to me? A lot of students don't come to see their professors. Let me tell you, I would go and sit in the hallway to see a professor. Like they would have office hours when I was in college, and there'd be so many people there, and I would just sit in line and wait for my turn. And I tried to sit really close to the door because I wanted to hear what other people were asking, right? To see if I could learn from that. And of course I did, just from hearing other people ask questions. So don't forget that I am a resource. And of course, I'm your biggest fan, and I want you to do well. Don't think of me as, inhibiting you from getting what you want, but instead, I give you the information. I can help you learn it. Okay? Let's see. Just so you know, my neighbors, I record these at home, right? My neighbors are getting their house painted this week. And so if you hear clang, 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 that is the ladder that they have that goes up to the second floor. And then if you hear people singing in Spanish, that would be the guys when they're working. So ignore the clang, clang, clang and the singing if it gets too loud. So here we go. Chapter 17, Urinary System Part 1. So we have 2 lectures on this. It's a tough system. Heck, has anything been easy in this class? No. The functions of the kidneys. Number 1, of course, is to form urine. It forms urine because it cleans the blood. So it filters the blood. It regulates blood volume. Remember, the blood is king, right? So we've got to keep the blood happy, regulate blood volume, ions, pH, and more. The kidneys eliminate nitrogenous wastes. You know that as urea from breaking down amino acids, right? They eliminate nitrogenous waste, toxins, drugs, and hydrogens. Remember, the hydrogens are all about the pH. And acid-base balance, which we will talk about in a couple lectures. We'll talk about the importance of the kidneys in keeping the pH stable of 7.35 to 7.45. So this is an anatomy review. The renal capsule on the outside, the cortex, the medulla, the pyramid, the minor. I expect you to know every word on here, okay? The nephron is where the urine is formed. It's called filtrate when it's inside, and then the urine is technically what we call the fluid once it reaches the minor calyx. The major calyx, the renal pelvis, and out the ureter. Blood vessels you need to know, renal artery, renal vein, all these things. Okay, so if you don't remember, take a little review. This is the part we're going to focus on, the nephron. So remember, the glomerulus is the blood coming in. So here's the afferent arteriole, glomerulus, and the efferent arteriole, and then we have the vasa recta and the capillaries going all around the nephron, where we can add and delete things. And then specifically the nephron part that I need you to remember the names of, the Bowman's capsule, I'm going to call it the glomerular capsule, and the proximal convoluted, that's funny that I messed up on the word convoluted, huh? Proximal convoluted tubule, and then the loop of Henle, and then the distal convoluted tubule to the collecting duct. All right? The micturition reflex, so that's what it means when you have to urinate. The bladder becomes full of urine, and I don't know if you remember this from anatomy, but there are stretch receptors there that are activated when there is between 250 and 400 milliliters of urine in the bladder. So get your water bottle out, and remember it's a 500 ml water bottle, so 250 is half of it. Not very much, right? Then of course you hold it or don't hold it. Some of you like to hold it all day. I don't know how you do that. Just keep holding it. The parasympathetic nervous system is involved in the reflex to contract the bladder. That should make sense, right? Rest and digest. And there are urinary sphincters, like the digestive tract, that have voluntary and involuntary control. So the internal smooth muscle sphincter, is that voluntary or involuntary? It's involuntary because it's smooth muscle. The external skeletal muscle is controlled, so that's voluntary. And I'll show you a little animation in just a second. This, like the sphincters of the digestive tract, are something the little kids can't control until they get myelination around all those neurons that innervate that smooth muscle or that skeletal muscle, and so that's potty training. That's what we do for little kids. The opposite of the micturition reflex is called the guarding reflex. The guarding reflex allows the bladder to fill by inhibiting the micturition center in the pons. Okay, so it's the micturition reflex and the guarding reflex. One makes it come out, the urine come out, the other lets it stay. Okay, let me show you the animation.
The micturition reflex involves impulses traveling from the urinary bladder to the sacral region of the spinal cord and from the sacral region of the spinal cord back to the bladder. It is coordinated by neurons in the spinal cord and can be influenced by signals from the brain. When the urinary bladder becomes stretched, there is an increase in the frequency of action potentials carried from the bladder wall to the sacral region of the spinal cord. In response, parasympathetic neurons from the spinal cord to the bladder are activated, and this causes the smooth muscle on the bladder wall to contract. The sensory signals to the sacral region of the spinal cord also stimulate ascending pathways to the pons and cerebrum, which results in a conscious desire to urinate. If urination is not convenient at the time, the brain sends impulses down the spinal cord to inhibit the micturition reflex. Impulses carried via somatic motor neurons keep the external urinary sphincter contracted, which also prevents urination. When urination is desired, signals from the brain stimulate the micturition reflex. The brain also decreases action potentials in the somatic motor neurons to relax the external urinary sphincter.
Alright, clinical stories. These little things are kidney stones. They look pretty small, but they cause a lot of pain when they try to come through the little teeny ureter and or urethra. Okay, getting through those tubes. So these are made of calcium oxalate. They get stuck in the ureters, and they are very painful. I don't know if you've heard of passing a kidney stone. So passing meaning it's got to make it through the ureters into the bladder, but then through the next tube, which is the urethra, and it's very painful. They can be treated with sonic waves or surgery. So with sonic waves, the idea is to break it up into small pieces so that it comes out without getting stuck. Surgery, of course, requires tubes to go inside and remove the pieces and get them pulled out. You can prevent kidney stones by drinking lots of water. So calcium oxalate that builds up with dehydration. So a lot of you don't drink enough water. You know that. I know some of you don't like water. That's fine. They make all these little powders you can put in your water. You can add lemon to it. You can add cucumber and mint and make it fancy like a spa kind of thing. So drinking lots of water, and I don't mean soda or Gatorade. Yes, those have water, and coffee has water. Milk, the number one ingredient of everything we drink is water. I mean just plain water, okay? I have a friend who gets kidney stones, and she can't believe the amount of pain that she has. And she's asking me, what can I do to prevent them from happening again? And I don't really know any big secrets. And luckily she hasn't had it again. But apparently once you get it, you're prone to getting them often. So maybe you can tell us some kidney stone stories when we have our discussion. Incontinence is what happens when the bladder is unable to hold the urine. And this happens as we get older. And you know this, right? Depends, right? The diaper is for adults, okay? This is very common as people get older. You will get older, okay? The aging decreases the strength of the sphincters. So, as we get older, people lose strength in their arms and their muscles and their legs, and even the strength of their heart, right? The diaphragm and the sphincters. So there are medications people can take. I've heard of the, it starts out, maybe in your 60s when you sneeze or cough, right? You've had that happen probably, where the urine comes out when you sneeze or cough. So there are meds that some people take. Exercise helps. Losing weight, very helpful, okay, because think about the weight, the pushing on the bladder, right? So when people lose weight, it helps that. Having timed bathroom breaks for older people, making sure they go every 2 hours, that way it doesn't get too full, so it doesn't leak out, okay? And I was at Costco recently, and two little old ladies pushing cart. I don't know if you've been to Costco, but they have really wide carts, like 2 kids can fit in the cart. And these 2 ladies were pushing it together, and I was like, oh, look at these old ladies shopping. They drove right over to the Depends. I didn't even know there were Depends there on the wall. They're looking at their coupons and how they're going to get a good deal. I thought, what a nice little date with your friend when you're older lady. Go to Costco, get the Depends, and I'll get a piece of pizza, right? The little treats that they have there. Probably got, some food. Anyway, I thought that was hilarious. I used to buy Huggies there for my kids, so it's just a circle of life, okay? 3 processes of urine formation are filtration, reabsorption, and secretion. Okay, so filtration is when we take the contents of the glomerulus and we squeeze out the little tiny small molecules. We'll talk about what can be filtered, what's small enough, and then we'll talk about what's too big and can't be filtered. So in this picture, they're showing the green dots coming through, too big to be filtered, but the blue ones can get across. And then we have reabsorption. So as we go, in this case, they just made the nephron tubing straight. But as you know, it curves and goes down and up and around. And this is reabsorption. So when we take the fluid in here now is called filtrate. When we keep it, or save it is what I call it, we save it, we put it back into the blood, that's called reabsorption. And then the blood vessels, the capillaries that surround this, sometimes they add extra stuff into the filtrate, and that's called secretion. At the end, excretion is removing it from the body, and now it's called urine when you get to the end. So this would be dumping into the minor calyx that we saw in the previous picture. Okay, so let's talk about each of these. Filtration is the movement of substances from the glomerulus to the glomerular capsule. 180 liters of filtrate is produced per day. So imagine a 2-liter soda bottle. Remember, you're not drinking it. 90 of those. If you could set up 90 of those in your room right now, they would just be stacked everywhere. That's how much filtrate you produce a day. And of course, you don't urinate that much. You couldn't even drink enough, right, to make that. The rate is then calculated into per minute, and that is 125 milliliters per minute. Okay, so looking at your little water bottle, that's about 1/4 of it is made every minute. And of course, we're going to save most of it. 20% of the plasma, so when you go through the glomerulus, 20% of that plasma goes into the Bowman's capsule, the glomerular capsule. That's a lot. So that's a lot of work for the kidneys. Second is reabsorption. This is the movement of substances from the renal tubing back into the blood. I call it saving. We save 178.5 liters each day. So we make 180. We save 178.5 5. So how much do we urinate out? 1.5 liters, right? And that's about right if you think about that. I mean, unless you haven't been drinking a normal amount for the day, right? But if normal day, about a liter and a half, it's about right. The third step is secretion. That's selective removal of substances from the blood to the renal tubules. And the last step is called excretion. Technically, it's not happening in the nephron, so the nephron only does filtration, reabsorption, and secretion. So elimination or excretion is getting things out of the body through the urine into the calyces, the renal pelvis, down into the ureters, and on down. So today we're going to talk mostly about filtration, and I think we'll get halfway through reabsorption. Then the next time we'll finish reabsorption and we'll add secretion to it. Okay, so about filtration, to review our anatomy, blood flows into the afferent arteriole, through the glomerulus, out through the efferent, and you can say efferent, whatever you want, arteriole. There are layers to get through. If you want to get from the capillary, the glomerulus, into the glomerular capsule, you have to pass through 3 layers. Okay, so this is showing you now detail with reminders that around the glomerulus there are these cells that are called podocytes. Okay, so it says podocytes of the visceral layer of the glomerular capsule. If this is the visceral layer, it's touching, remember visceral is touching, this over here is the parietal layer. So here it says parietal layer of the glomerular capsule. If you look more closely, these fit together like putting your fingers, if you take your hands right now, put your fingers, lace them between each other in your 2 hands, you can see how things can still get through your fingers, right? There's spaces between them. That is part of the filtration membrane. So there are 3 parts, that's one. There's also the capillary endothelium here, has little holes called fenestrations or fenestra. There's also a basement membrane, here it is, and then those slits that we just talked about between the podocytes. Okay, so these are the spaces here, the filtration slits between the little pedicels, which are the parts that stick out. They're called podocytes because they look like little feet sticking out. Okay, there's my clicker here. Filtration, so I show a picture of, what are these things in the corner? These are paper coffee filters, yeah. So filtration membrane has the fenestra or fenestrations, whichever word you like, fenestra of the glomerulus. You say that word. Say glomerulus. It's hard. You get stuck on it. Glomerulus capillary. Second is the basement membrane of the capillary. Third is the visceral layer of the Bowman's capsule. So those are the podocytes with spaces called filtration slits. What kinds of things are filtered? Well, water is small, of course. Salts are small. Glucose is small. Even little amino acids or small di or tripeptides could get through. Okay? But a cell is too big. A white blood cell is too big. A red blood cell. Proteins are too big. So if we see proteins in the urine, that would be a bad sign. If we see cells in the urine, that would be a bad sign, unless the cells have come from the urethra or the ureter or the bladder. Then it would be okay. But if it comes from the blood, it's not supposed to be there. Okay, big things can't get through. That fluid now is called filtrate. Okay, it's not called plasma, it's not called urine, it's called filtrate. So let's fill this out, and it should be blank on your page. I want you to just listen to me and then pause the show and fill out the form. Okay, so just listen. I want to talk about what pushes the forces. This is physiology. Okay, the forces that push or inhibit filtration. Okay, so the first is the blood pressure in the glomerulus. Don't write this down, just listen. Glomerular blood pressure favors filtration, right? So fluid wants to move out because of the pressure inside the glomerulus, pushing it out, right? The amount of the pressure is measured, remember, in millimeters of mercury. It is at 55. So remember, your blood pressure measured in your brachial artery is 120/80. 120/80. By the time we get to the glomerulus, down in the kidney, what happens to the pressure? It's going down, it's down, so we're now at 55. Okay? Just listen. There are proteins in your blood that want to keep, remember this, keep fluids in. So remember the albumin? This is called the glomerular colloid osmotic pressure. And it opposes filtration. Okay, it wants to hold fluids in. And it has a pressure of 30 millimeters of mercury, trying to keep fluids in. So remember this in-out thing that we did before? We're doing it again. And there is already fluid in the glomerular capsule, right? There's filtrate already there. That makes a pressure called the capsular hydrostatic pressure. Hydro means water, right? So this is fluid in the capsule. That is preventing the stuff from being filtered. Okay, the fluid is kind of in the way of the new fluid from getting through. So that opposes filtration, and you get a pressure of 15 millimeters of mercury, preventing or opposing filtration and keeping fluid in. Okay, so this is the same thing we did before with this in-out stuff. You add it up, 55 pushing out, 30 pushing in, 15 pushing in, and what do you get? You get what's called the net filtration pressure. It favors filtration, and the grand total is 10 pushing out. Okay, so take a second, pause the show, fill out your form, so it looks like this. Make sure you get it, and that you can do this math, and that you memorize these three forces. Okay, it should be kind of easy since we did this. I mean, it's never easy, but it should be easier since we already did this before. Okay, pause me. So this next topic is about regulating that glomerular filtration rate. So what was the number? Do you remember? What's the GFR, the glomerular filtration rate? It was 180 liters per day, but we need to know the milliliters per minute. So the GFR is 125 milliliters per minute. Okay, so we control it. You remember extrinsic and intrinsic control, right? The sympathetic nervous system constricts the afferent arteriole. If you constrict the afferent arteriole, what will happen to the glomerular filtration rate? Well, what will happen to those numbers? What will happen to the blood pressure inside of the glomerulus if you decrease the flow that comes into the glomerulus? The blood pressure will drop, and then what will happen to the GFR, to the glomerular filtration rate? It will decrease, right? We will be practicing this on one of the physio-X that you do. Okay, you'll get to change the afferent arteriole, make it smaller, make it bigger, change the efferent, make it larger, make it bigger, and you'll be able to see how the GFR, or the amount of urine coming out, really changes. Intrinsic control. If the blood pressure drops, the afferent arterioles will vaso-blank to keep glomerular blood pressure normal. So if blood pressure drops, in order to keep it normal, okay, if that 55 drops, the afferent are going to vasodilate, right? They have to say, hey, we're not getting enough blood into the glomerulus. Please open so we have more flow, more blood, more volume, more pressure. Trying to keep it normal. If the blood pressure is too high, the afferents are going to vaso what? They're going to vasoconstrict to keep the glomerular blood pressure normal. Okay, so it's also, we're talking about keeping the blood happy. The kidneys are another one we try to keep really happy because we want the glomerular filtration rate to be working properly so that we get rid of toxins. If it shuts down, then we get too toxic and poisonous, right? Okay, so this we're done. PhysioX is where you're going to practice all this stuff. Let me give you a question to work on. It says, explain what would happen to glomerular filtration rate and urine volume in these patients. 1. A patient with liver disease. Think how the liver affects albumin levels. So remember, the liver makes albumin. And a patient with kidney stones. Think how the stones affect the renal tubules. Okay? These are hard questions. Pause the show, think about it, and then we'll do the answer. Okay, the patient with liver disease. Let's go back. Let me go back. This one. Patient with liver disease. I told you the clue was it would change this colloid osmotic pressure. Do you think a person with liver disease is making more or less albumin? Probably making less albumin. So if they have less albumin, they have this number higher or lower than 30? Probably less, right? Probably lower. So let's say this was 25. Can I change my pointer to be a pen? How about that? See if I can write like this. So let's say this 30 is no longer 30 and instead it is 25. Okay? It's kind of hard to write with my mouse. If this is 25 because there's less albumin, what happens to the 10? So we now have 55 out, 25 in, 15 in. Do we have more or less coming out? We have more coming out, right? So we have 15, right? 15 coming out. So what, if this is 15, what does that mean the GFR is doing? Is the glomerular filtration rate higher or lower? GFR goes up. So a person with liver disease has an increased GFR. They urinate more. It's one of the symptoms that they have is frequent urination. And that's because they don't make enough of the albumin to hold the fluid in. Okay? Let's see, the other one was the kidney stones. Kidney stones, can I go back up to the picture? Is there another picture? This one. Kidney stones block. Oh, back. Kidney stones, they block right here. Okay? If this blocks here, what happens to the pressure in the Bowman's capsule? To this fluid. What happens to this pressure in this fluid inside the Bowman's capsule? The pressure goes up. Okay, so now, going along, see if I can go back. We had 55 out. That stays the same. Unless, of course, the person was bleeding, right? If they had hemorrhage, that would change. If they had liver problems, that changed. But this is changing the hydrostatic pressure. We just said it would go up. So let's say now this is 20. Okay? So we have 55 out, 30 in, 20 in. So the net filtration pressure for the person with kidney stones is how much? It's only 5. So what happened to the GFR? Did it decrease or increase? Decreases. So this person is not urinating, not getting it out because there's a block, really, a blockage, and that hydrostatic pressure has gone up. Okay, did you get either of those right? It's hard. Got it? Think, yeah, okay. Good. This is the next topic about reabsorption. So reabsorption is, and I need my pointer back. Let me bring my pointer arrow, okay? Reabsorption is the movement from, in this picture, the lumen of the tubules is at the top of the page. This is the proximal tubule cell, the yellow one, and then we have this intermembrane, or this interstitial fluid here, and then we have the capillary. Okay, so reabsorption goes from the top of the screen to the bottom of the screen. And what we want to talk about is how that stuff and what stuff gets across. Okay, so today we'll get through half of this. In this case, sodium has to cross in order to be reabsorbed, okay, because we filter it small. Water, salts, we said, amino acids, glucose. Okay, so in this case, let's talk about the sodium. It first has to cross this membrane, which is called the apical membrane. Then it has to go inside of the proximal tubule cell. Then it has to cross another membrane, which is called the basolateral membrane. Then it has to go into the interstitial fluid. Then it has to cross into the capillary. Okay, so this is a lot of work in order to save it. And it's being very particular. You see, it's not just letting stuff come flying across. It actually has to go through membranes and needs transporters and all kinds of stuff. So let's do the sodium. There is lots of sodium in the lumen. So it comes across through a co-transporter, which is a protein in the membrane that we learned about at the beginning of school. It's a sodium glucose co-transporter. So there's a lock and key fit here. There's a spot to fit the sodium, a spot to fit the glucose. So the sodium comes in, and the glucose kind of got a free ride to go with it. The sodium then comes through your friend, the sodium-potassium pump. And of course you need ATP. You have potassium coming the opposite direction. Sodium now coming out through this side. When it comes through, then it just goes through one of the little holes, fenestrations of the capillary. Okay? To get from the lumen to the proximal tubule cell, I wrote this. You won't see this in the picture in the book. This is my writing. We have a high concentration of sodium to a low concentration of sodium, and this is called passive diffusion, right? But to get from low to high concentration, you have to do what? You have to do active transport. So here we have the pump to do that. We take the low concentration, we pump it out so that it goes into the capillary, and that's low to high. So that's primary active transport. Okay? Let's see, what else do I want you to see here? What else? I just want to say something. High to low, low to high. The glucose comes along, and then it just has a facilitated diffusion here. Remember, it's just a protein in the membrane that goes from high to low. Let's see what else I want. Let's go to next slide and see what it says. The reabsorption in the proximal convoluted tubule. Sodium is reabsorbed there. Lots of sodium, the high to low, low to high thing. So see the picture. 80% of kidney energy is used for active transport across the basolateral membrane. Okay, 80%. Kidney uses a lot of ATP. And 80% of the ATP it uses is to transport that sodium with the sodium-potassium pump. So if we look through the nephron, we start at the proximal convoluted tubule. You'll see that 65% of the sodium that came through filtration is now reabsorbed here in the proximal convoluted tubule. I write PCT to save space on my slides, otherwise my slides get too full. You won't see it in the book as PCT, and people don't call it PCT, just like they don't call it AP for action potential. This is just my wording, okay? 25% is reabsorbed in the loop of Henle, and we'll talk about that more in a minute. And then 10% is reabsorbed in the distal convoluted tubule. And some of the sodium is not reabsorbed. It comes out in the urine. Okay, if you have a high salty meal, you don't want to reabsorb all of it, so you won't keep it all. So these are this distal convoluted tubule one. We can really adjust that 10% to be more or less if we want. Okay, next we have other ions and molecules like potassium is reabsorbed, and this is not the potassium that I just showed you in the picture. That potassium was going to be secreted, okay, because it was going in the opposite direction. But we do pump potassium to reabsorb it. Glucose is co-transported with sodium from the lumen to the tubule cell. We reabsorb 100% of our glucose unless we have diabetes that's uncontrolled. Okay, because we have glucose to make ATP, and if there's any extra, we don't let it go like sodium. We keep it. We store it as glycogen, or we store it as adipose, right? We store extra glucose as lipids to be used for energy at a later day. Chloride ions follow the sodium and potassium passively. So if sodium is reabsorbed or potassium reabsorbed, chloride just gets a free ride. And water follows the salt by osmosis. So where there is salt, water follows. So if we reabsorb a lot of salt, we're going to reabsorb a lot of water. Fun fact, an average human drinks about 16,000 gallons of water in a lifetime. 16,000 gallons. I don't know. Maybe more, right? I don't even know. I think that's a lot, but I don't know. Maybe it's not that much. Reabsorption in the loop of Henle. Okay, so that was reabsorption, proximal convoluted tubule. Now we did the loop of Henle. The loop of Henle has what we call the medullary countercurrent system. This helps to concentrate the filtrate through the descending limb and then dilute through the ascending limb. Now I know a lot of students feel like that's a waste of effort to go down and back up again. All you do is concentrate and then dilute, but it allows for a lot of reabsorption of both the salts and the water. So we're looking at the picture here. We have 300 milliosmoles of filtrate. This is the filtrate coming in, and this is the concentration of the solutes, 300. As you go down, you reabsorb. What do you reabsorb here? Water, water, water. So what happens to the concentration as you go down? Bigger, 600, 800, 1,000, 1,200, 1,400. So by the time you get down to the bottom of the loop of Henle, you've now totally concentrated your filtrate. And then you go up the other side, and you have a pump here. The X is referring to a pump, where you pump out sodium. And of course, who goes along for the ride? Cl minus, right? Chloride, sodium, sodium chloride, sodium chloride. And so what happens to the concentration? 1,400, 1,200, 1,008, goes down, 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 till you get to the top of the loop of Henle where you're at 1,000, or what number is that? 100. The thing is, this pumping of sodium puts salt here. Who likes the salt? The water does. Yeah, so the reason the water comes out is because of osmosis, and the reason it wants to come out is because there's all this salt here. Okay, so the salt coming out of the ascending limb actually attracts the water through osmosis from the descending limb. Turns out this side, the descending limb, is only permeable to water, so the salt can't even get out over here. And this side has all these pumps in it, and that doesn't let water out on this side, only the salt. Okay, so we go down and up, and at the top, I want you to remember this number, 100. Okay. So reabsorption in the loop of Henle. Oops, wrong way, sorry. If we fill this out, the limb, whether it's impermeable or permeable, and how. So the descending limb is impermeable to Na+, Cl-, and potassium. It's permeable to water. And how does the water get out? Osmosis. The ascending limb is impermeable to water, permeable to sodium, chloride, and potassium. How does that get out? Through the sodium-potassium pump. Okay, so this is a lot of ATP required here too. Okay, so we were talking about these needs of the kidney. It uses a lot of energy. Okay, so this is called the vertical osmotic gradient. How we had all that salt at the top and bottom. So the sodium and potassium are pumped out of the ascending limb. This makes the osmolality high at the loop, so the 1,400, and then water leaves the descending limb by osmosis. I think we've said this. I already said this. I probably didn't need this slide. I feel like I already said that. Okay, so what I want you to see now is these numbers again. We started with the filtrate that had 300, proximal convoluted tubule, 300, 300, go down, 400, 600, 800, 1,200, 1,400, back up, the salt leaves, 1,200, 600, 400, 200. There are numbers also here in the interstitial fluid too. And those numbers you can see are the same, right? They're the same. So that's what causes, of course, the water to come out. I want you to see that at the top we have 100 going through the distal convoluted tubule to the collecting duct. And once you get to the collecting duct, you can change the number. You can have the 100 that's there come all the way out, or you can change it. Let's see, the 100 could come out, or you can reabsorb some water here and maybe have 300 milliosmoles come out. Or reabsorb, say you're really dehydrated. Really dehydrated, you need to save water. The urine that comes out, really concentrated. It's a bright yellow, not a lot of volume, and say 1400 is going to be the solute concentration. All this water comes out through the collecting duct because of what hormone? Which is the hormone that saves water? Anti-diuretic hormone. Yes, so the one you've talked about so long, so many times, now we know exactly where it's working. There are water channels here that are opened when anti-diuretic hormone is released. Okay? So our last slide, ADH increases the permeability of collecting duct to water by stimulating aquaporin channels. I love that word, aquaporin, right? The pores, they're holes for aqua, for the water. When you have ADH, you know this, you increase water reabsorption, you save water, you increase blood volume, increase blood pressure. You're so good with this guy. The urine can be 100 milliosmoles, which is very dilute, or as I said, if you're really dehydrated, it would be bright yellow, not a lot of volume, and the concentration would be really high at 1400 milliosmoles. The result is increased blood volume and increased blood pressure. We know that. And ADH, of course, release is controlled by the hypothalamus and osmoreceptors. So the blood is king. Okay, so the osmoreceptors check the blood constantly to make sure the osmolality is good. Okay, this is a tough lecture, huh? Got a lot to do. Watch it again and again and again and again. We're done.